Welcome to my video on renal physiology. I will be focusing on FRCA Primary syllabus and SOE exam oriented. I am aware that the college has issued an examiner report considering cancelling SOE in the future. But given that the college has to announce at least 12 months before making any changes to the exam format, the SOE will still exist for at least the coming year. I also find preparation of MCQ and SOE not mutually exclusive. MCQ asks a lot about the fine details, while SOE emphasizes on understanding the topic and deliver an answer in an organized way. Doing revision as if you were preparing the SOE actually helps you understand the topic and integrate concepts as a whole. For SOE exam, there are 2 examiners marking each part independently. 2 marks are given for a pass, 1 mark for borderline performance, and 0 mark for fail. For exam purpose, here are a few tips for answering SOE. Listen carefully to what you are being asked. Do go straight for the money instead of hovering irrelevant details. If you get asked about definition, just give a neat answer. Always start answering with the keyword, key sentence, or punchline. Imagine your examiner listen to your repeated answer. He can barely focus on what you say. An SOE is an impression marking without boxes to tick. First impression is important. Let's go through hot questions in SOE. These questions either appear in past paper or in exam books. Let's try to go through them and find how to deliver an answer beautifully. So what is our normal renal blood flow? The renal blood flow is 1200 ml per minute, or 500 or 600 ml to each kidney, which equals to 25% of our cardiac output. You don't have to mention the rule of 5 unless you get prompted, and definitely not explaining the autoregulation without being asked. The rule of 5 means 1/5 of cardiac output, 500 ml per minute to each kidney, 500 ml per minute per 100 gram to cortex. 1/5 of this go to outer medulla, and further 1/5 of that goes to inner medulla. So next question, how is the renal blood flow regulated? The renal blood flow is mainly regulated by triple-glomerular feedback and autoregulation mechanism. Triple-glomerular feedback and autoregulation are the keywords. Triple-glomerular feedback involves sensor, transmission, and response. Again, give headings to your answers. Macula densa is situated in the wall of distal convoluted tubule and detects sodium and chloride delivery. A reduced delivery of sodium or chloride will stimulate the release of adenosine into the glomerulus and make the afferent arteriole vasoconstrict and reduce renal blood flow. Autoregulation is controlled by myogenic and metabolic mechanism. Afferent arteriole vasoconstrict or dilate in response to the transmural pressure. It is also mediated by the metabolic substance including prostaglandin, thromboxane A2, nitric oxide, and endothelin. It is also under the influence of autonomic nervous system sympathetic stimulation to vasoconstrict, as well as hormonal effect of angiotensin II. In this way, you start with keywords, followed by headings, and then into the details. That's how you score 2 in SOE. Next question, can you compare and contrast renal and liver blood flow and their regulation? Compare and contrast is another favorite type of questions in SOE. When you do your revision, don't just focus on single system, but try to integrate your knowledge from every system and compare. My suggestion here is to draw a table. Makes it much easier to compare things in exam, and you can talk it through while you write. The cerebral, coronary, renal, and liver blood flow and autoregulation mechanism are commonly asked in exam. So try to memorize these numbers and understand the implication behind. For example, coronary vessels are tiny and therefore only receive 5% of the cardiac output, but it extracts up to 60% of oxygen to support its high metabolic rate. On the other hand, the highly vascularized liver receives up to 30% of cardiac output as a buffer, but only extracts 7% of oxygen as many zone 2 and 3 anaerobic metabolism doesn't require oxygen. Understanding the implication makes it easier to memorize the figures. Now the examiner asks, how do we measure renal blood flow? So what are the keywords? We calculate the renal blood flow by plasma clearance of pH using the fixed principle. So the pH and fixed principle are the keywords. Fixed principle states that the amount of substance in blood that flows to organ equals to the amount that flows out, if the organ does not produce or degrade that substance. About 90% of pH that enters the kidney through the renal artery is filtered and excreted in urine, and only 10% enters the renal vein, which is assumed to be negligible. So we assume the amount of pH entering the kidney equals the amount in urine. As the renal blood flow is a function of renal plasma flow, we use the equation to derive renal blood flow from the renal plasma by dividing it by 1 minus hematocrit. There are two assumptions here, first being pH in renal vein is negligible, and secondly, being peripheral venous blood sampling equals to the renal artery pH concentration. Right, next question. What is the normal GFR and what governs the rate? So the normal GFR is 125 mL per minute. The GFR is determined by two factors, filtration force by Starling equation and glomerular permeability. Starling force states that the net filtration rate is a function of the forces favoring filtration and those opposing it, and can be described by the following equation. The GFR is dependent on differences between the hydrostatic and oncotic pressure within the glomerular capillaries and the Bowman's capsule. Compared to normal vascular beds, pressure drops less in the afferent arterioles, allowing a high glomerular capillary pressure of 50 mm mercury. As the Bowman's capsule is a blind endotube, there is a hydrostatic pressure which opposes the pressure gradient at 10 mm mercury, as well as the initial capillary oncotic pressure of 25 mm mercury. You could consider drawing the glomerulus in the Bowman capsule if that helps you explain. Integrate your knowledge to other body systems. Filtration force not only applies to renal glomerulus, but also applies to systemic and pulmonary vasculatures. They share the same equation with different pressure gradients. Permeability is another determining factor, which is determined by mesangial cells, molecular size, and charge. Structure and headings. Mesangial cells lie between the capillary endothelium and the basement membrane, with contractile function to reduce the surface area for filtration. They are under the influence of neurohormonal effects, including the angiotensin II and prostaglandins. Graham's law states that the filtration rate is inversely proportional to the diameter of molecules, unless the molecules are small enough to be freely filtered. Finally, the basement membrane is negatively charged, so any negatively charged molecules will be filtered slower. So, how do we measure GFR? This is a trickier question because there is no perfect way to measure an accurate GFR. Being said that, GFR can either be calculated by fixed principle using clearance of an ideal substance, or estimated using prediction formula. As previously mentioned, the fixed principle states that the amount of substance in blood that flows to the organ equals to the amount that flows out, if the organ does not produce or degrade that substance. So we need a substance that is freely filtered at the glomerulus, but not excreted nor reabsorbed. The amount in the urine must equal to the amount filtered and equal to the amount that goes into the kidney, which we could measure. We can't use the pH as we do in the renal blood flow measurement, because a portion of it is secreted instead of filtered. That would overestimate the GFR. The CNG and the MDRD equations are some validated formula to estimate the GFR, but both have their limitations. So what is the most ideal substance to be used for GFR measurement? Inulin is the closest to the perfect. It is freely filtered, not reabsorbed, secreted, nor metabolized, but it is expensive. Creatinine is the most commonly used agent clinically, but it requires 24 hours urine collection, and a small amount is secreted through the tubules. Urea is not reliable, given up to 50% can be reabsorbed, and it could be affected by factors like diet and tissue breakdown. So next question, how does the kidney concentrate urine? The kidney concentrates urine by osmotic gradient created by the countercurrent mechanism. So the keywords are osmotic gradient and countercurrent mechanisms. I would suggest everyone to practice drawing the countercurrent tubules. It helps a lot in explaining the complicated mechanism. The important annotation of the diagram include the descending limb permeable to water, while ascending limb being impermeable. The sodium chloride actively reabsorbed from ascending limb and water passive osmosis into the interstitium. Also, annotate the extreme osmolality on both ends of the tubule, 100 milliosmol in the cortex and 1200 milliosmol in the inner medulla, because it tells you how dilute or concentrated our urine can be. The efficiency of osmotic gradient is maximized by the help of FRCA, which runs in opposite direction against the filtrate, which carries away the sodium chloride reabsorbed from the ascending limb to maintain a steep concentration gradient. But it is unnecessary to draw the FRCA, which might be too complicated. The ultimate purpose of such a complicated mechanism is to create an osmotic gradient to allow water to be reabsorbed in the collecting tubule. The collecting tubule is relatively impermeable to water, but under the action of ADH or vasopressin, released from the posterior pituitary under the influence of baroreceptors and osmoreceptors, aquapore will be inserted into the collecting tubule and open channels to allow water to be reabsorbed. I will talk about the osmoregulation in my next video. So next question, how does the kidney handle glucose? Again, a diagram can help to explain. Nearly all glucose that is filtered will be reabsorbed and occurs almost entirely in the proximal tubule. In normal plasma concentration, the amount filtered is directly proportional to the plasma concentration. Entry is linked to the secondary active transport sympod mechanism with sodium. The transport molecule is saturable. This means, above a threshold concentration, further absorption is not possible, and the filtered glucose is lost in urine. The threshold concentration is around 22 milliosmol. The tubular maximum is the highest rate of transport of a particular molecule. So how do electrolytes exchange takes place in tubules? This is a very broad question involving too many details. It is rather unlikely that you will be asked to explain them all. You might be asked to explain one of the proximal or distal tubule, if you get asked at all. But for teaching purpose, I could quickly go through them and highlight the important ones. So for proximal convoluted tubule, which involves primary and secondary active transport, facilitated diffusion, and paracellular movement. For primary active transport, it involves the sodium-potassium ATPase on the basal membrane and the sodium-hydrogen exchange on the apical membrane. Secondary active transport uses ionic gradient created by ATPase to release energy and allow transport of other solutes. This is therefore indirectly linked to the ATP breakdown. The most important ionic gradient is sodium. It happens to glucose reabsorption with sodium. For facilitated diffusion, is the term given to describe transport via the ion channels and uniporters, like calcium here. Paracellular movement is the movement occurring through the spaces between cells and nephrons. They may be driven by concentration, osmotic or electrical gradients, like the calcium here. So for Loop-Henle, here is the ascending limb of the Loop-Henle, which we have mentioned before. The most important exchange through the apical membrane occurs with the sodium, potassium, and chloride cotransporter, and is extruded through the sodium-potassium ATPase on the basal membrane. This leads to a buildup of sodium chloride in the interstitium. So for distal convoluted tubule, the primary active transport with sodium-potassium ATPase on basolateral membrane and secondary active transport with sodium chloride cotransporter in apical membrane, together with facilitated diffusion by sodium and calcium ion channels. The collecting duct involves principal cells and intercalated cells. They play an important role in exit-based regulation, which I will explain in other videos. But for water reabsorption, it is facilitated by aquaporin channels through the action of ADH. I wouldn't bother practice drawing all these diagrams, but I might memorize the most important channels for each of these tubules. So thank you for watching my video on renal physiology. Make sure if you haven't subscribed my FRCA Primary Resources Platform website, please do subscribe it, and you'll be notified when we have new videos to be rolled out. Thank you very much.